Biochemical composition, structure, functions, and genome organization of mitochondria. Mitochondria are membrane-bound organelles present in cytoplasm of all eukaryotic cells that produce adenosine triphosphate, the main energy molecule used by the cell. Popularly known as the powerhouse of the cell, they play a major role in breaking down nutrients and generating energy-rich molecules for the cell. Many of the biochemical reactions involved in cellular respiration take place within the mitochondria. The term mitochondrion is derived from the Greek word mitos and chondrion, which means thread and granules like, respectively. It was first described by a German pathologist named Richard Altman in the year 1890. Biochemical composition of mitochondria. Composition of mitochondrial membrane is just like the plasma membrane, that is phospholipids and proteins. Protein is present on two surfaces and a biomolecular layer of lipid in between the two. Outer membrane contains more cholesterol and is higher in phosphatidyl inositol. There are 0.5% RNA and small amount of DNA and respiratory enzymes which help in respiration like cytochrome oxidase, reductase, transaminase, coenzyme 1, octanoxidase, fatty acid oxidase, etc. Phospholipids are also important for enzyme function, since their addition to purified enzymes also increases activity. Mitochondrial lipids are largely phospholipids, lecithin being prominent, but triglycerides, phosphatidic acid, and cholesterol are also present. All the three major components of food, that is carbohydrates, protein, and lipids, degraded in cytoplasm enter mitochondrial Krebs cycle and undergo oxidation. These are some of the mitochondrial enzymes like monoamine oxidase, fatty acid coenzyme A ligase, nucleoside diphosphokinases, adenylate kinase, NADH cytochrome C reductase, etc. Mitochondrial DNA. Mitochondria contain one or more molecules of DNA which are circular in shape, highly twisted, and measure about 5 microunit in length. It resembles bacterial DNA which is also circular in shape. A large proportion of DNA may occur in looped form. Replication is not synchronized and continues throughout the cell cycle, and it is not in coordination with cyclic nuclear DNA replication. DNA synthesis is slower in mitochondria than in DNA. Mitochondrial DNA codes for intrinsic ribosomal RNA, transfer RNA, ribosomal proteins, and few other proteins, regulating enzymes and structure. Many of the mitochondrial proteins, especially enzymatic proteins, are coded by nuclear RNA, nuclear DNA. Mitochondrial RNA. It is different from RNA of nuclear origin as it is resistant to ribonuclease enzyme. It is synthesized since mitochondria on DNA template. Three species of mRNA, namely 23S, 16S, and 4S, have been isolated so far. Structure of mitochondria. The mitochondrion is a double-membraned, rod-shaped structure found in both plant and animal cell. Its size ranges from 0.5 to 1.0 micrometer in diameter. The structure comprises an outer membrane, an inner membrane, and a gel-like material called the matrix. The outer membrane and the inner membrane are made of proteins and phospholipid layers separated by the intermembrane space. The outer membrane covers the surface of mitochondrion and has a large number of special proteins known as porins. Cristae. The inner membrane of mitochondria is rather complex in structure. It has many folds that form a layered structure called cristae, and this helps in increasing the surface area inside the organelle. The cristae and the proteins of the inner membrane aid in the production of ATP molecules. The inner mitochondrial membrane is strictly permeable only to oxygen and ATP molecules. A number of chemical reactions takes place within the inner membrane of mitochondria. Mitochondrial matrix. The mitochondrial matrix is a viscous fluid and that contains a mixture of enzymes and proteins. It also comprises ribosomes, inorganic ions, mitochondrial DNA, nucleotide cofactors, and organic molecules. The enzymes present in the matrix play an important role in the synthesis of ATP molecules. This is a structure of mitochondria showing its different sections and parts. Functions of mitochondria. The most important function of mitochondria is to produce energy through the process of oxidative phosphorylation. It is also involved in the processes like regulates the metabolic activity of the cell, promotes the growth of new cells and cell multiplication, helps in detoxifying ammonia in liver cells, plays an important role in apoptosis or programmed cell death. Responsible for building certain parts of the blood and various hormones like testosterone and estrogen, helps in maintaining an adequate concentration of calcium ions within the compartments of the cell. It is also involved in various cellular activities like cellular differentiation, cell signaling, cell senescence, controlling the cell cycle, and also in cell growth. Genome organization of mitochondria. Mitochondria play a central role in cellular energy provision. The organelles contain their own genome within a modified genetic code. The mammalian mitochondrial genome is transmitted exclusively through the female germline. The human mitochondrial DNA is a double-stranded circular molecule of 16,569 base pairs containing 37 genes coding for 2 RNAs, rRNAs, and 22 tRNAs and 13 polypeptides. The mitochondrial DNA encoded polypeptides are all subunits of enzyme complexes of the oxidative phosphorylation system. Mitochondria are not self-supporting entities but rely heavily for their functions on imported nuclear gene products. Cis-acting mitochondrial DNA sequences have been characterized by sequence comparisons, mapping studies, and mutation analysis both in vitro and in patients harboring the mitochondrial DNA mutations. Characterization of trans-actin factors has proven more difficult, but several key enzymes involved in mitochondrial DNA replication, transcription, and protein synthesis have now been biochemically identified, and some have been cloned. Plant mitochondria have a complex and peculiar genetic system. They have the largest genomes as compared to organelles from other eukaryotic organisms. The presence of autonomous plasmids of essentially unknown function further enhances the level of complexity. The physical organization of the plant mitochondrial DNA includes a set of subgenomic forms resulting from homologous recombination between repeats with a mixture of linear, circular, and branched structures. Recombination appears to be an essential characteristic of plant mitochondrial genetic processes, both in shaping and maintaining the genome. Genome structure of mitochondria. Thank you.